Hello everyone, I am Dr. Richa Sahu, working as an assistant professor in Amity Institute of Biotechnology, Amity University, Chhattisgarh. I welcome you to the Swayam course of genetic engineering and its recent advancement. So today we will be talking about gene expression, an introduction to gene expression. In the previous lectures, you have studied about gene silencing, its introduction, RNA interference. In the subsequent lectures, you will be studying much detail about gene expression. So starting with the gene expression, what is gene? So gene basically is a DNA segment containing the biological information, hence coding for a RNA or a polypeptide chain. It seems complicated. If you see, there is a myriad of organism in this whole white planet. Each organism has a specific genome which specifies it. It tells us how the organism is developing, how it is maintaining, how it is surviving the environment. So this is, every organism has a specific genome. So higher organism, including humans, the genome that we have is DNA, deoxyribonucleic acid. However, the lower organisms like viruses have RNA genome or ribonucleic acid genome. So there are two types of genome, and in genome you have gene, which is a DNA segment containing the biological information which codes for a product which is required for the survival of the cells. It may be RNA, it may be a polypeptide or protein molecule. When the gene expresses, this phenomena is known as gene expression. So gene expression is basically the process where the genetic information stored in the gene or the DNA molecule is converted into a specific products like RNA and protein. It is a series of events by which the biological information carried by a gene is released and made available to the cell. The process by which information from a gene is used for the synthesis of functional gene product like RNA and polypeptide chain or protein. So, gene expression study is a very fundamental process or very fundamental study to understand how the organism develop, how the organism maintain, and how the organism sustain the environment they are in, how they adjust in the environment. By, like for example, if how the color of the eye is blue, how the color of my eye is brown, how the hair that you have is curly, how the hair of another person is straight. What is defining it? There is a gene for each trait, which when expresses, it is expressed in the form of the phenotype. So the gene expression, if in totality, if I say, it is the process by which the information that is stored in the genome or gene is used and processed for the synthesis of a functional gene product for its application or its various function, and the functional gene product can be RNA or polypeptide chain or protein. In many cases, this functional product of a gene is protein, sometimes it is RNA. For example, if we talk about gene expression, let's take an example. Let's take an example of Mendel flower color. If Mendel flower color gene, any gene which is coding for the color of the flower, let's say for example, it gives the instruction to the protein that helps to make the colored pigments on molecules. Because of that, the flower becomes of that color. Like for example, if the Mendel or if the flower color or the petal color has to be violet, there is a gene encoding a protein. This gene will result in the synthesis of mRNA. Then this mRNA will result in the synthesis of protein, a specific protein which will result or which will produce this violet pigments. Because of that violet pigments, the phenotype that you see would be a violet colored flower. This is how the gene expression works. What is the importance of gene expression? Why do we study it? Why we have so much fuss around what is gene expression, why are we studying it? So gene expression is a fundamental process in the biology, and it is very, very crucial to understand the functioning of all the living organisms. As I told you, how they function, how they maintain, how they survive. It is responsible for the development, growth and maintenance of the organism. So during the gene expression, there are two types of gene. One is protein coding gene, one is non-protein coding gene. During the expression of protein coding gene, the information flows from DNA to RNA via transcription. Via transcription, an mRNA is formed, and from the mRNA, via translation, a protein or a polypeptide chain is formed. This is a series of gene expression. But for the non-protein coding gene, the gene that means that does not specify any functional protein, they are just RNA. So in case of non-protein coding gene, they are still transcribed, the transcription will happen. It will lead to the formation of RNA like rRNA or tRNA, but these two RNAs will not be translated into polypeptide. That does not mean they are not expressed, they are not important. So these are known as non-protein coding gene, which lead to the formation of rRNA and tRNA. For either type of gene, the process of going from DNA to a functional product, I'll repeat, for either type of gene, coding or non-coding, coding, protein coding or non-protein coding, the process of going from DNA to a functional product, may it be protein or RNA, it is known as gene expression. So again, before going to the expression of gene, we need to understand one very basic concept of central dogma of molecular biology. What is central dogma? Central dogma is the conversion of information stored in the DNA to RNA via transcription and from the RNA to protein via translation. Alright, so this is central dogma. The central dogma was proposed by Francis Crick in 1958. This basically describes the flow of genetic information within the biological system from DNA to mRNA and then to protein. The central dogma of molecular biology or molecular genetic, as you can call it, consists of three main processes: replication, transcription and translation. Moving ahead with gene expression. So gene expression basically consists of two steps: transcription and translation. That means the synthesis of RNA and the synthesis of protein. These two processes comprise in totality a gene expression. But can you study RNA and protein without DNA? Like for example, there is a stress condition. A particular type of protein is required to sustain that stress condition. That can be anything. The organism requires a particular type of protein. How will you get a protein in a large number? You need mRNA. You need mRNA in large number. And how will you get mRNA? From the DNA. That means you need multiple copies of DNA. You need multiple copies of that gene, which will lead to the formation of mRNA, and from mRNA via translation protein. So you have to study DNA replication in gene expression. So what is DNA replication basically? It is the process by which the cell makes an identical copy of its DNA molecule or genetic material. It is a very crucial step in cell division, as it ensures that each daughter cell receives a complete set of genetic instruction. What happens in cell division? There are 4 phases of cell division. When the DNA replication happens, the DNA replication begins in the S phase of cell cycle. In S phase, via DNA replication, 2 identical copies of DNA is created. During the G2 cycle, the cell makes its final preparation for the cell division. It prepares for the cell division and replicates the organelle so that the organelle can be divided or the process in every cell, every daughter cell. After the G2 phase, the cell division occurs in the mitosis or the M phase. After the cell division, each daughter cell receives an identical copy of DNA created during S phase. So DNA replication is very important. After DNA replication, cell cycle happens and each daughter cell gets an identical copy of the genetic material which has been synthesized during the S phase of cell cycle. Talking about DNA replication, the first step. Then we will move on to transcription and translation. See, you will be studying about DNA replication in detail in subsequent lectures. Today we will be talking about an overview of DNA replication, transcription and translation. How it works, because we are talking about gene expression. So, starting with DNA replication. So, before going into the steps of DNA replication, let's talk about what is the starting material that you need for DNA replication. You need a DNA molecule. because you need a template for the synthesis of DNA. So, what will you need? A template DNA on which the second copy of DNA will be formed. The next thing that you need is DNA polymerase enzyme. What does it do? It helps in the synthesis of the DNA molecule. It elongates the daughter DNA chain. You need helicase enzyme. क्या करता है helicase enzyme? It opens the double stranded DNA. It opens the double stranded DNA so that the strand can act as a template for the daughter DNA molecule. Next you need is a single stranded DNA binding protein, SSB protein. What does it do? It stabilizes the single stranded DNA. What happens when the helicase enzyme opens the DNA, double stranded DNA? It tends to go back to the double stranded form. They go back to each other. So to stabilize the single stranded DNA that is prepared by the helicase enzyme, you need single stranded DNA binding protein to stabilize it. Next you need is what? The primase enzyme and DNA primer. What is that? To initiate the DNA replication, you need a primer molecule, you need a RNA primer molecule. Why? And for the synthesis of RNA primer, you need primase enzyme, which is also known as RNA polymerase. In the subsequent lecture that you will be taught, you will be studying about primase and it is called RNA polymerase. Why? Because the DNA polymerase cannot start the DNA replication de novo from the scratch. It cannot do that. So it needs a RNA primer, and to synthesize RNA primer you need primase enzyme. So these are the starting material that you need for DNA replication. Of course, the DNTPs. Talking about steps of DNA replication, it starts with the helicase enzyme opening the double stranded DNA and making the replication fork, and SSB molecule stabilizing the single stranded DNA. Now the primase enzyme makes the RNA primer and it attaches to the strands of DNA, the DNA template. It is the template, the RNA primer attaches to both the template. And the DNA, now what happens, the RNA polymerase or the primase enzyme makes the RNA primer, attaches to the template, and the DNA replication is initiated by the DNA polymerase enzyme. Now the next step is elongation. The DNA replication, the elongation basically is from the 5' end to 3' end. The elongation cannot happen from 3' end to 5' end. So in 3, because there are 2 strands in the replication fork like this. So one is the lagging strand, another one is the leading strand. The leading strand is the strand where the elongation of the DNA, new DNA, is taking place from 5' to 3' end. Another one is the lagging strand because it is few nucleotide lagged behind the leading strand, because the DNA molecule, the new daughter DNA is made in small fragments of Okazaki, known as Okazaki fragments. The next step is finally the termination. So when the DNA is replicated, the ligase enzyme join it, the RNA primer is separated, the DNA polymerase 1 joins the Okazaki fragment, and you get the daughter 2 identical copies of your DNA. So this is just an overview of DNA replication. It's a very complicated process that happens in the cell, and it is very much regulated. It is very much regulated, all the regulation, how it happens, all the steps in detail, what attaches first, what leaves the DNA first, all these things we will be studying in detail in the subsequent lectures. After the DNA has been synthesized, now the next step is the transcription. So basically, transcription is the first step of gene expression. So what is DNA transcription? It is the process by which the genetic information contained within the DNA is rewritten into messenger RNA by RNA polymerase enzyme. The mRNA then exits the nucleus after the synthesis or the transcription, which takes place in the nucleus. This DNA replication and transcription both takes place in the nucleus. And after the RNAs has been synthesized in the nucleus, they leave the nucleus, and the mRNA which leaves the nucleus becomes the basis for the protein synthesis via translation. So, DNA transcription, I will repeat again, is the process by which the genetic information contained within the DNA is rewritten into messenger RNA or mRNA by RNA polymerase. Via transcription, another two types of RNAs are also synthesized using the DNA template, that is rRNA and tRNA. So, there are three steps in DNA transcription, that is synthesis of RNA: initiation, elongation and termination. In the initiation step, the RNA polymerase enzyme binds to the template DNA at the promoter region. Okay, before going to the steps of the transcription, let's talk about what are the prerequisite of, what are the things that are required by the cell for transcription. You need a antisense DNA strand, a template on which the RNA polymerase binds. You need RNA polymerase enzyme, because you don't need anything else, you don't need a primer, because RNA polymerase can start or initiate the RNA synthesis de novo. So, now you have a DNA helix, double stranded DNA helix. The RNA polymerase enzyme binds to the DNA helix at the template strand where the promoter region is present. This is how the transcription is initiated. The beauty about RNA polymerase is it melts the double stranded DNA helix. It opens it a little bit so that the transcription can happen. It melts the hydrogen bond, opens the double stranded DNA at the promoter region where it is binding and initiate the transcription process. Next step is elongation. Like DNA replication, the elongation of transcription takes place from 5' to 3' end, and the RNA polymerase moves throughout the template, melting the hydrogen bonds, opening the DNA strand, and once the DNA polymerase moves, they again become double stranded. So, in the elongation step of RNA, like DNA transcription, RNA synthesis I would say, it again takes place from 5' end to 3' end. The next step is termination. When the RNA polymerase reaches the terminal end of the DNA, it attaches to single strand, but the DNA is double stranded. It detaches itself from the template and releases the RNA that has been formed. This is just an overview of transcription. There are so many transcription factors that are in the scene, there are so many other enzymes that are in the scene, the regulatory factors are there. You will be studying about that in very detail in the subsequent lectures. So, after the three steps like the termination, what you have is the RNA molecule, all the three types of RNA molecules, the RNA polymerase get detached and the DNA again becomes double stranded. After the RNA has been detached from the RNA polymerase, you have the RNA molecule. In case of eukaryote, there is processing of the mRNA. Why? Because in the eukaryotic genome, you have introns and exons. Alright, what are introns is the non-coding area, exons are the coding area. So, when the mRNA is formed, it has both introns and exon area. Unprocessed mRNA that is prepared in the eukaryotic DNA transcription cannot be used as a template or cannot be used for protein synthesis. Therefore, after transcription in eukaryotes, there is post-transcription modification. There is a processing of mRNA where the introns are removed. What you have is a mRNA with only exons. Now, the mRNA, the processed mRNA that we have goes for translation. What is translation? Translation is basically the synthesis of protein molecule using the information in the mRNA that has been synthesized via transcription. After transcription, an mRNA molecule is ready to direct the protein synthesis. The process of using information in the mRNA to build a polypeptide chain in the translation. During translation, a cell reads the information in the mRNA and it uses it to build a protein. Is the last step in central dogma where the information in the mRNA is read and the information is used to build a polypeptide chain or a protein. So, before going into the steps of translation, there are few things that should be known which are very important in translation step is codon, because it is recognized. The codon is very important in translation. So, what is codon? Just a small overview. A codon is a DNA or RNA sequence of 3 nucleotide or a trinucleotide that forms a unit of genomic information encoding a particular amino acid or signaling the termination of the protein synthesis. So basically what is codon? It's a DNA or RNA sequence of 3 nucleotide long which is encoding a particular amino acid. There are total 64 codons. 61 codons specify an amino acid. 3 codons are used as a stop signals. So these codons only signal the termination of the protein synthesis or the translation process. So there are 3 steps of protein synthesis or translation. The first step is initiation. In the first step, small ribosomal subunit bind to the mRNA and the initiator transfer RNA attaches to the start codon, that is AUG. What happens? The ribosome subunit doesn't bind anywhere in the mRNA. It binds to the particular area where the start codon is present, AUG. AUG codes for methionine. When the small ribosomal subunits bind to the mRNA at the start codon and the initiator transfer RNA, which has an anticodon, attaches to the place where the small ribosomal subunit is binding to the mRNA. This is how the translation is initiated, by binding of the ribosome on the mRNA where the start codon AUG or methionine is present. The next step is elongation. The ribosome moves along mRNA matching the transfer RNA anticodon with mRNA codon. So mRNA has different nucleotides, and the set of 3 nucleotides is known as codon. The ribosome binds to the start codon, that is AUG methionine, and it keeps on moving and it recruits the different types of transfer RNA carrying an anticodon which is specifically coding a particular amino acid. This happens, and there are many types of tRNAs present in the cytoplasm that time. It will recruit that tRNA which is carrying the anticodon which is matching to the codon which is present where the ribosome is there after AUG, and these tRNA keeps on adding amino acid to the growing polypeptide chain, making the polypeptide bond. The next step is termination. Translation stops when a stop codon is reached and the polypeptide is released. What happens? There is mRNA and the ribosome smaller subunit is bind to the mRNA, where the initiation occurs at the AUG, the start codon. It moves 3 codons, it moves and it recruits the transfer RNA, and transfer RNA is carrying one amino acid. Alright, like for example UCG is one codon which is an anticodon for serine amino acid. That means the opposite of UCG. The transfer RNA having the antisense codon or the anticodon of UCG where the ribosome is present will attach to the mRNA and transfer this serine amino acid to the growing polypeptide chain, making the polypeptide bond. And this continues, the elongation step continues, it keeps on repeating. The ribosome, the smaller subunit, keeps on recruiting different tRNA and it keeps on moving on the mRNA till it finds the stop codon. There are 3 types of stop codon. When it reaches the stop codon, it immediately stops and terminates the addition of another amino acid. So this is how the protein or the polypeptide chain is made, and the transcription and translation together, it is known as gene expression. So the gene expression and the regulation of gene expression is different in prokaryotic cell and in eukaryotic cell. There's a difference in gene expression and regulation in both type of cells. For example, talking first about the prokaryotic gene expression and regulation. The first thing is in prokaryotic cell, the transcription and translation, they take place in coupling together. That means in prokaryotic cells, the transcription, that is the synthesis of RNA, and the translation, that is the synthesis of polypeptide chain or protein from RNA, are coupled. There is no difference. Means once the DNA is replicated, it immediately goes into transcription and RNA is formed, and immediately that mRNA is used for the translation, that is for the synthesis of protein, which doesn't happen in eukaryote. We'll talk about eukaryote in forthcoming slides. Next is the regulation. Talking about the regulation of cell expression in prokaryote, they are regulated by operons. One of the example is lac operon or lactose operon. What are the operon system? These prokaryotic genes are often organized into operons, which are clusters of genes with related functions that are transcribed as a single unit. This allows for the coordinated regulation of gene expression. The operons include a promoter, where the RNA polymerase bind, an operator, a regulatory element, and the gene cluster, the gene themselves. So, this consists of the operon system which are used for the regulation of gene expression in prokaryotes. The next is there is no post-transcriptional processing. As I mentioned when I was explaining the transcription, that in prokaryotes the mRNA does not undergo extensive post-transcriptional processing. It is typically polycistronic, that means containing multiple coding regions, and that can be immediately translated. Another region is the absence of introns. They don't have a non-coding region unlike the eukaryotic genome. So, they don't need a post-transcriptional processing which happens in eukaryotes. The next is rapid response. It's very, very important. Prokaryotic gene expression can respond rapidly to the changing environmental conditions. Because there are very fewer steps in the process and less regulation. The genome of the prokaryotes are very simple. The response to the environmental condition is very rapid. As I told, there are very few regulatory steps. Next, if we talk about the eukaryotic gene expression regulation, the transcription and translation processes are separate. One reason in eukaryotic cells, the transcription occurs in the nucleus, while the translation occurs in the cytoplasm. Both the places are separated to the presence of double membrane bound nucleus that houses the DNA. The transcription and translation occurs at two different places, though they cannot occur in coupling as happened in the prokaryotes. The next is the presence of introns and exons. After transcription, eukaryotic mRNA undergoes post-transcriptional modification. The eukaryotic gene often contains introns and exons. Introns are the non-coding regions within the gene, while the exons are the coding regions. The pre-mRNA is synthesized with the introns and exons both and undergoes splicing or the post-transcriptional modification to remove introns before becoming the mature mRNA. And this mature mRNA then takes place in the translation. There are multiple levels of regulation in eukaryotes. The regulation is very complicated. The eukaryotic gene expression is subjected to extensive regulation at multiple levels, including transcriptional regulation, enhancers, promoters, presence of transcription factors, post-transcriptional regulation, RNA processing, splicing, stability, transport, regulation, and post-translational modification where the protein is folded, phosphorylated, etc. So, the gene regulation in case of eukaryotic cells is of multiple levels and very extensive and complicated. In eukaryotes, the promoter structure is very complicated. It's very complex. The eukaryotic gene have more complex promoter regions compared to prokaryotes, often involving enhancers and silencers that can be located far from the gene itself. And finally, eukaryotic cells or eukaryotic gene have a slower response. Unlike prokaryote, they have a rapid response. The eukaryotic gene expression tends to be slower to respond to environmental changes due to the multiple layers of regulation and the spatial separation of the transcription and translation. This sums up the overview of the gene expression. So, in this lecture, what we talked about, we started with the central dogma so that we can understand what gene expression is. We talked about what is gene expression, alright. What are the steps in the gene expression? We talked about DNA replication, because if the multiple copies of protein is required, you require multiple copies of mRNA. So, for the multiple copies of mRNA, you need multiple copies of DNA. So, we first talked about DNA replication, an overview of DNA replication, what exactly happens, what are the steps. Then we moved on to the transcription. There we talked about the steps of the transcription, what are the machinery that is involved in the transcription phenomena or the process. Then we talked about there is a modification of the mRNA that is synthesized via transcription, what is the machinery. And then we moved on to the translation, that is the synthesis of protein. In translation, we talked about codons, because it is very important the main machinery which takes part in the translation process is ribosome. And ribosome recognizes the codon on the mRNA. So we talked about codon, that there are 64 types of codon, 61 codes for amino acid and 3 are the stop codons. Then we talked about the steps of the translation: initiation, elongation and termination. And next, the next segment of this lecture was the difference between the gene expression and regulation between the eukaryotic and prokaryotic cells, where we talked about the complexities of eukaryotic gene regulation expression system. However, the prokaryotic system is comparatively simpler. That is why they have a rapid response towards the external stimuli or the change in the environment. The eukaryotic system have a slower response to the external environment. We have a very complex regulation system. They have a comparatively simpler regulation system. So this completes the overview of gene expression. In the subsequent lectures, you will be talking in detail about the DNA replication, about the translation, transcription, regulation of gene expression. These are the topics which will be dealt in much more detail in the forthcoming lectures. ठीक है।